Today, we're going to talk about a really powerful technique scientists use to find the hidden stories inside our genes. It's a method that takes these enormous piles of data and turns them into actual, meaningful biological discoveries. Okay, let's jump right in.
And that problem, believe it or not, isn't that we don't have enough information. No, it's actually the complete opposite.
I mean, after just one experiment, a scientist might be staring at a list of thousands of genes that have changed. On its own, that list is just, well, it's just noise. The real challenge is finding the signal, the actual story that's buried inside all of that data.
So let's really put this into perspective. Imagine you're comparing cancer cells to healthy cells. An experiment can easily spit out a list of all the genes that are more active or less active in those cancer cells. But then what? You're left with this huge, overwhelming list of gene names. What do you do with it? How do you turn that list into actual knowledge?
To really get how scientists solve this, let's use a simple analogy. Just forget about genes for a second and look at this shopping list. At first glance, it just looks like a random bunch of items. But is it really?
You know, the amazing thing is how our brains do this automatically. We don't just see a bunch of individual items, we instantly see that flour, sugar, eggs, and chocolate chips, they all kind of belong together, right? They're all baking ingredients. And by noticing this enrichment of baking stuff, we can figure out the hidden story. Someone is about to make cookies.
And that exact same logic, that same aha moment, is precisely what scientists are trying to apply to that overwhelming list of genes. They're looking for the biological version of baking cookies.
And this whole process has a formal name. It's called enrichment analysis. It's basically a really clever statistical method that asks a simple question. Within my huge list of genes, is there a surprisingly high number of them that all do a similar thing? Is there a group of baking-related genes that tells me a single, coherent story?
So this is the key takeaway right here, the direct translation. Your shopping list, that's your list of interesting genes. The grocery store aisles, like the baking aisle or the dairy aisle, those are the known biological pathways. And the story of baking cookies becomes the biological story, like discovering that your cancer cells have a super active pathway for something like cell growth. This is the game-changing step that takes you from a simple list to a profound insight.
All right, so to actually pull off this kind of biological detective work, an analyst needs a few key tools in their toolkit. Let's take a look at what they are.
First up, you obviously need your list of interesting genes. That's our shopping list. Second, you need some background context, what scientists call a gene universe. Think of it this way. To know if finding chocolate chips on a shopping list is unusual, you first need to know if you're in a grocery store or a hardware store, right? The gene universe is the grocery store. It's the complete list of all the genes that could have possibly shown up, which gives us the context we need to see what's truly special. And last, you need your reference databases. These are the digital libraries that work like your aisle categories. And two of the biggest, most important libraries for this kind of detective work are called Go and Keg. They're these massive public databases that basically catalog everything we know about what genes do.
The easiest way to think about it is like this. The Gene Ontology, or GO database, is like a dictionary. It tells you what a single gene does, its function, its biological process, like cell division. KEG, on the other hand, is more like a recipe book. It groups genes together into known pathways, the step-by-step instructions for a complex function, like how a cell turns sugar into energy.
Okay, so all this theory is great, but let's see how it all works in a real-world example. We're going to look at a classic dataset that compared two different types of leukemia, ALL and AML. The goal was to find the biological story that makes them tick differently.
So here was the game plan. First, the scientists found all the genes that were way more active, we call that upregulated, and all the ones that were way less active, or downregulated, when comparing the two cancer types. Next, they defined their gene universe as all 5,421 genes they measured in the experiment. And then they took their lists of interesting genes and basically cross-referenced them with the Go and KEG databases to see what popped up.
And this is what they started with. After their first pass, they had a list of 597 interesting genes. But hold on, isn't this just our original problem all over again? We're still just looking at a long, uninterpreted list.
So how do we finally make sense of it all? Well, the output of an enrichment analysis can still be a pretty dense table of stats and names. The next, absolutely crucial step is visualization.
But here's the next challenge.
Even after running the analysis, you get a table that looks something like this. It tells you, okay, 86 biological processes were enriched here and 9 pathways there. I mean, that's a ton of information, but it's not a clear story just yet, is it?
We have to transform this wall of numbers into something our brains can actually process easily. A visual map of the biological story that's unfolding.
And that's where these amazing things called network graphs come in. In a visual like this, each circle, or node, is a biological process that the analysis found. The colors tell the story. Red nodes are for processes that were way more active, and green nodes are for processes that were less active. And the lines? They just show you how all these different processes are connected to each other, revealing the entire biological network at a single glance.
And this brings us to the ultimate payoff of this entire technique. Because it's about so much more than just numbers and pretty graphs, it's about real, tangible discovery. This quote right here just captures the whole thing perfectly. Enrichment analysis is really a form of computational storytelling. It gives us a structured way to look at our data and ask, okay, what are you really trying to tell me?
And this is why it all matters so much. By finding these hidden stories, we can start to understand what's actually going wrong in a disease like cancer. We can pinpoint the specific pathways that are broken, and that gives scientists critical clues for developing brand new therapies. It turns a boring list into a roadmap for research.
Of course, it's also worth remembering that the specific method we've walked through is really just one example. There are many different flavors of enrichment analysis out there and lots of other tools that scientists use, but they all share this exact same fundamental goal.
So we'll leave you with this thought. If we can use this kind of method to start decoding leukemia, what other complex diseases, everything from Alzheimer's to diabetes, are just sitting there, waiting for us to find and read the incredible stories hidden deep inside their genetic code?